Good morning, everyone. 5 minutes, wait other students, and start our lecture. I'm glad to see you again. Let's begin. The topic our lecture today, individual development of the organism. Plan of our lecture. The first question, ontogenesis and its periodization. Next question, gametogenesis. Next, fertilization, cleavage, gastrulation, primary and final organogenesis, and post-embryonic development period. This lecture will last 2 lectures. Today we consider ontogenesis, gametogenesis, fertilization, and cleavage. What is ontogenesis? Ontogenesis is a process of individual development of organisms. There are 3 periods of ontogenesis. First period, pre-embryonic period, which includes gametogenesis and fertilization. Second period, embryonic period, which includes cleavage, gastrulation, primary and final organogenesis. And third period, post-embryonic period, begins with birth or hatching and ends in death. And the first question, pre-embryonic period, includes gametogenesis. Gametogenesis is a process by which the diploid germ cells undergo a number of chromosomal and morphological changes to form native haploid gametes. Animals produce gametes directly through meiosis in organs called gonads, testes for male and ovary for female. Males and females have different types of gametogenesis. Spermatogenesis for male, sperms are produced in testes, and ovogenesis for female, eggs are produced in ovary. Gametogenesis, spermatogenesis, and ovogenesis includes the following stages: reproduction, the first stage; second stage, growth; next stage, maturation; and last stage, formation. Stage formation is absent for oogenesis. So spermatogenesis has 4 stages, oogenesis only 3 stages. On this slide you see process spermatogenesis. Consider firstly process gametogenesis, consider firstly spermatogenesis. So you see spermatogenesis and oogenesis on the slide. Let's begin with spermatogenesis. The first stage, reproduction. You must know what occurs on each stage and what name of cell on each stage and how many set of chromosomes on each stage in the cell. The first stage, reproduction. What occurs? Cells divide by mitosis and their number increases. During spermatogenesis, on this stage, cell is called spermatogonia. You must remember names of cell on each stage. Reproduction, cell is called spermatogonia. Spermatogonia divide by mitosis and their number increases. Before mitosis, each spermatogonia has 2n4c because before each cell division, doubling DNA occurs, and each chromosome consists of 2 chromatids. After mitosis, each spermatogonia has 2n2c because during anaphase of mitosis, chromatids move to opposite poles, and each cell will have only 1 chromatid in 1 chromosome. Second stage, growth. Spermatogonia slightly increases in size, accumulates some nutrients, and turn into spermatocyte 1 or a primary spermatocyte. So on the second stage, state of growth, spermatogonia transforms in primary spermatocyte. Third stage, maturation. The main process during this stage is meiosis. You must remember that meiosis consists of 2 cell division, first meiosis and second meiosis, and during meiosis, 1 diploid mother cell gives 4 haploid daughter cell. cells, genetic materials of which different from genetic materials of mother cell. And you see on this slide that primary spermatocyte undergo meiosis. After first meiosis, primary spermatocyte divide on two daughter cells and names of this cell secondary spermatocyte. То есть one primary spermatocyte divide on two secondary spermatocytes. Each secondary spermatocyte has 1N, 2C. Because the first meiosis is reduction cell division. On this stage, secondary spermatocyte undergo second meiosis, and each secondary spermatocyte divides on two daughter cells again, and names of this cell are spermatids. Each spermatid has 1N, 1C. So, during spermatogenesis, during state maturation, one primary spermatocyte divides on 4 daughter cells. These cells are called spermatids. You can see two names of cells on this stage, you must remember. Secondary spermatocytes after first meiosis and spermatids after second meiosis. Genetic material spermatids are different because during meiosis crossing over occurs and there is independent assortment of homologous chromosome during anaphase 1. And last stage of spermatogenesis, formation. During this stage, spermatids transform in spermatozoa or sperm. Set of chromosomes remain same spermatid, 1N or 1C. So, spermatogenesis consists of 4 stages: reproduction, growth, maturation, and formation. The main process during reproduction, mitosis. The main process during growth and accumulating nutrients. The main process during maturation, meiosis. And during formation, morphological changes. Oogenesis. Oogenesis is a process of formation eggs. Oogenesis includes only 3 stages: reproduction, growth, and maturation. State of formation is absent during oogenesis. On the first stage of reproduction, oogonia divides by mitosis, divides by mitosis, and their number increases too. And you can see a lot of oogonia in ovary. Next stage, growth. Oogonia is no longer divided, going through interphase doubling the nacreous, and accumulates many nutrients. They will be needed for the further development of the embryo. A cell of this state is called primary oocyte or oocyte 1. Next state, maturation, the main process is meiosis II. Primary oocyte undergo the first meiosis, and primary oocyte divide on two daughter cells with set of chromosome 1N to C. But what different from spermatogenesis? The peculiarity of ovogenesis is that during meiosis the cytoplasm divides unevenly, and primary oocyte divides on two different daughter cells, one big daughter cell and one small daughter cell. Big daughter cell is called secondary oocyte. Small daughter cell without cytoplasm, without nutrients, which has only nucleus and thin layer of cytoplasm around nucleus, is called polar body. After first meiosis, primary oocyte divide on two different cells. Big cells, secondary oocyte, and small cells, polar body. After second meiosis, the cytoplasm divides unevenly, and secondary oocyte divides on two daughter cells again, one big and one small. And big daughter cell is called eggs or an ovum, a small polar body too. Polar body, which forms after first meiosis, divide on two polar body. So we have that one primary oocyte during state of maturation divides on four daughter cells, but only one egg and three polar bodies. It's process of oogenesis. And I want to show you short film from YouTube about oogenesis. 1 minute.
The ovary is the site of the development of the oocyte. Within the ovaries, follicles develop. Each follicle houses an oocyte, which matures simultaneously with the follicle. The primary follicle houses an oocyte encapsulated by a coating called the zona pellucida. It also contains a layer of granulosa cells that surrounds the oocyte. The secondary follicle contains many more granulosa cells, and an accumulation of follicular fluid that is secreted by granulosa cells. This fluid coalesces in the follicle to form the antrum. A mature follicle is much larger than a secondary follicle, and the antrum occupies a greater portion of the follicle. It is within the mature follicle that the first meiotic division of the oocyte is completed, to produce a secondary oocyte and the first polar body. The secondary oocyte receives most of the cytoplasm from the cell division. However, the nuclei and the chromosomes are divided equally. The polar body degenerates. The mature follicle ruptures at ovulation to release the secondary oocyte and a corona of granulosa cells into the uterine tube. The second meiotic division cannot be completed unless the secondary oocyte unites with the sperm cell. As a result of this division, a second polar body is formed, and the oocyte and sperm nuclei combine to form a zygote.
And you see this process on this slide. One primary oocyte divide on secondary oocyte and polar body. After that, polar body divide on two polar body, and secondary oocyte divide on eggs and polar body too. On this movement, state of formation during oogenesis is absent. I repeat. Structure of the sperm cell. On this slide you see structure of spermatozoa or a sperm. Each spermatozoa has a head, you see, neck with this region, and tail. Head encloses a compact nucleus, you see brown on this slide, and is kept by a vesicle called acrosome vesicle, red on this slide, which is derived from the Golgi complex. The acrosome contains enzymes that take part in penetration of protective layers surrounding the egg. And on the surface of membrane located a lot of receptor. The neck contains a proximal centriole. The tail consists of three patches. Intermediate or a midpiece, main patches, where you can see flagellum, and terminal patches, end patches without flagellum, not these patches, end patches. Axial fiber is principal component of the whole tail. In the intermediate part, there are many mitochondria which supply energy to him. And main structure of tail is flagellum. You must remember that flagellum consists of axoneme, and wall of axoneme consists of 9 duplet of microtubules and 2 non-pair microtubules in the center. Structure of flagellum we considered last lecture. Structure of ovum of Sea-Akin. Oocyte or ovum is female germ cell. It is the largest cell, maybe 130 to 150 micrometer in diameter in human body, which cannot move. Each is covered with a few tunics from outside the eye. The first tunics, corona radiata, purple on this slide. You see a lot of follicular cell in corona radiata. Corona radiata tunica granulosa consists of numerous follicular cells which give their process to ovum through the previous tunic. Their functions are protection and nutrition of the oocyte and regulation of its maturation. Next layer, orange or a yellow on this slide, zona pellicida. Zona pellicida is a special chemical membrane which contains much glycoproteins, glycosaminoglycans. glycoproteins present by three types: ZP1, ZP2, ZP3. ZP3 is receptive for spermatozoa. ZP2 and ZP3 prevent polysperm. And on the surface of oocyte, you can see plasma membrane is called ovolemma. In the cytoplasm of oocyte, you can see nucleus with genetic materials. In the cytoplasm may be a lot of granule of nutrients, for example, yolk. And under plasma membrane, you can see a lot of small vesicles, cortical granules. Secret of cortical granules is prevent polysperm. Common terms for eggs. Egg has animal pole. The pole of egg, the yolk is least concentrated. The cytoplasm without yolk, or animal hemisphere, hemisphere of the egg where animal pole is located. Vegetal pole, the pole end of egg where yolk is the most concentrated, or a vegetal hemisphere, hemisphere of egg where vegetal pole is located. Type of egg. Eggs are classified according to amount or a distribution of yolk. According to the amount of yolk, the eggs are arranged into the following types: aletital without yolk, for example, eggs of mammal; oligoletital, small amount of yolk, eggs of Amphioxus, for example; mesoletital, moderate amount of yolk, for example, eggs of frog; polyletital, large amount of yolk, for example, eggs of bird and reptile. According to the distribution of yolk, there are next type of eggs. Homoletable egg. The yolk is equally distributed throughout the cytoplasm, for example, Amphioxus. Centroletical egg. The yolk is concentrated in net-like structure around the nucleus, for example, insects. And teloletical egg. The yolk is deposited in vegetal pole, for example, amphibians, birds, reptile. Next process, fertilization. Fertilization is the process of fusion of male and female gametes and the formation of a diploid zygote. There are three stages of fertilization. The first stage, short information, distant interaction of gametes. Sperm and egg cells produce hormones. Hormones activates sperm and provides their movement to egg. Next stage, contact interaction of gametes. This stage is characterized by acrosomal reaction of the sperm and cortical reaction of the egg. As a result, the nucleus of sperm penetrates to the egg and the fertilization envelope, which prevents the penetration of other sperm, forms around the egg. And third stage, formation syncharion. The nuclei of the sperm and egg unite and form a diploid zygote. And consider this state more carefully. Distant interaction of gametes. Sperm and egg cell produce hormones. Their joint action activates sperm and provides their movement to egg. And you can see two processes on this state. Capacitation. Capacitation means special activation of spermatozoa, which occurs with them in female reproductive organs. Only after such changes they begin to move forward. Taxis. Taxis is active passage of spermatozoa. Chemotaxis, chemotrapism, taught to the oocyte, which produce special chemical substance, hormones. Rheotaxis against the fluid flow in uterine tube. Contact interaction of gametes. This stage is characterized by acrosomal reaction of the sperm and cortical reaction of the egg. Contact state of fertilization includes penetrate of the corona radiata, penetrate of the zona pellucida, penetrate of the ovarium. Penetrate of the corona radiata. On this state, 200 to 50 spermatozoa surround the egg and join into corona radiata. The egg begins to rotate too, and corona radiata cells disperse. And you see this process on this slide. With this follicular cell of corona radiata disperse. Penetrate of the zona pellucida. Only one spermatozoa can penetrate of the zona pellucida, what you see on this slide. When this spermatozoa contacts with the P3 glycoproteins, each receptor of this zona, acrosomal reaction begins. What is acrosomal reaction? You see on this slide. The acrosomal reaction is release of acrosomal enzymes. This reaction allows spermatozoa to penetrate of zona pellucida and then to come in contact with the plasma membrane of the oocyte. And you see binding of sperm to zona pellucida, receptor of spermatozoa join with the ZP3 receptor of oocyte, acrosomal reaction occurs, enzymes from acrosome is released, and destroyed zona pellucida, and membrane of spermatozoa join with plasma membrane of oocyte. and penetrate of the overlemma. After addition of plasma membranes of the sperm and egg is matched, the spermatozoa nuclear mitochondria, the patch of axonema, enter in the ooplasm. But the spermatozoa plasma membrane is left behind on the oocyte surface. As soon as the spermatozoa has entered to the oocyte, the egg responds by two ways. Cortical reaction, the first phase, and second, resumption of the second meiotic division. Cortical reactions. This reaction includes a rise in iron calcium that stimulate cortical granules to release their contents outside the egg. I say, under plasma membrane of each oocyte, you can see special granule, cortical granule, with special chemical substances. And during cortical reaction, by exocytosis, content of cortical granule are outside on plasma lemma. And this chemical substance join with receptor on the surface of oocyte. This change cause formation of a fertilization envelope that function as a block to polyspermia. When new spermatozoa come to oocyte, receptor of spermatozoa cannot join with receptor on the plasma membrane because this receptor has other chemical structure. And next way, resumption to the second meiotic division. You see on film now. One of the daughter cells is known as the second polar body. The other daughter cells is the definitive oocyte. То есть, when spermatozoa penetrate zona pellucida and go to oocyte, second meiotic division occurs, and secondary oocyte divide on two daughter cells, polar body and egg. Each chromosomes for human 22 plus X arrange themselves in a vesicle nucleus known as the female pronucleus. And next stage, formation of syncyrion. The nuclei of the sperm and egg unite and form a diploid zygote. Syngamia is the process of nuclear fusion. A male pronucleus unites with a female pronucleus. You see on this slide. At the moment of their fusion, such unicellular organism is named syncyrion. As a result, a zygote is formed, which consists of, for human, 46 chromosomes. So you see nuclear envelope of female and male pronucleus disintegrate and chromosomes fuse. And you see cell for human on the state of metaphase. And the first division of zygote is beginning. We considered pre-embryonic period. Pre-embryonic period includes, remember, gametogenesis and fertilization. Next period, embryonic period. Is the first stage of embryonic period, cleavage. Cleavage is the process of repeated rapid mitotic cell divisions of the zygote unicellular structure to form the blastula multicellular structure. The produced cells are named blastomeres. During this stage, the size of the embryo does not change. The blastomeres become smaller with each division, smaller and smaller. The type and pattern of cleavage differ from species to species. Continuous division to form a ball of 32 cells called morula. The morula continuous division to form the hollow blastula with up to several hundred cells. The cavity of the blastula is the blastocoel. So, result of cleavage, formation of blastula. Blastula has wall, is called blastoderm. This wall consists of a lot of cell, blastomeres, and cavity in the center of blastula is called blastocoel. And you see on this slide, blastula consists of a wall blastoderm consisting of individual cells, blastomeres, and the cavity, blastocoel in the center. It's process formation of blastula for Amphioxus. Process formation of blastula for frog, for example. To consider more carefully. The pattern of cleavage is influenced by two factors, the amount of yolk and the formation of mitotic spindles. Cleavage can be complete or a holoblastic and incomplete or a meroblastic. Holoblastic cleavage can be uniform if all blastomeres of equal size and uneven when blastomeres of different size. Holoblastic cleavage can be synchronous if blastomeres are divided at the same time and asynchronous if blastomeres are divided at the different time. So, if you see complete cleavage, you must say complete, for example, uniform, synchronous, complete uneven, asynchronous. Always will be 3 words about complete cleavage. Meroblastic cleavage can be superficial and discoidal. Superficial cleavage, the nucleus divides into several ones which migrate toward the surface which surround by cytoplasm, for example insect, and discoidal cleavage localized in dominal disc, for example birds. And consider this process more carefully on the example of organism. Amphioxus. Amphioxus has egg which are legletidal and homoletidal. Type of cleavage is complete holoblastic, uniform and synchronous. As a result, coiler blastula is formed. And you see coiler blastula of Amphioxus. Each blastocoel is filled with mucopolysaccharide. The blastocoel is centric. And you see thin layer of blastoderm from small blastomeres. Amphibians. Egg of amphibians is mesolatial and telolatial. Pole where egg located is called vegetal pole. Pole without egg is called animal pole for egg. Type of cleavage is complete holoblastic cleavage, but uneven because blastomeres of different size. You see small blastomeres of animal pole and big blastomeres on vegetal pole, and asynchronous because blastomere divide in different time. Type of blastula, amphiblastula. Cytoplasm without yolk, with animal pole of egg without yolk, and cytoplasm without yolk divides faster than cytoplasm with yolk. So on animal pole you can see a lot of small blastomeres. These blastomeres are called micromeres. On vegetal pole you can see a big blastomeres with yolk. These blastomeres are called macromeres. And you see, it forms two types of structurally different blastomeres. Micromeres on animal pole without yolk and macromere on vegetal pole with yolk. In practical class, you will draw blastula of amphibians and consider under microscope if offline. Birds. Eggs of birds polylatial and telolatial. You can see eggs of birds and cytoplasm without yolk located on the surface of yolk like small disc. This egg has only small disc cytoplasm without yolk on the surface of yolk. A lot of yolk in this cell. So, type of cleavage incomplete. And only cytoplasm without yolk can divide. So, cleavage discoidal. This cytoplasm divide and form one layer of cell on the surface of yolk. This layer of cell is called discoblastula. And blastocoel is space between yolk and discoblastula. The embryo forms a disc of cell called a blastodisc on the top of the yolk. Blastocoel is very, very small. Insect. Egg of insect is polylatial and centrolatial. Type of cleavage is incomplete superficial cleavage. And type of blastula will be periblastula. single layer of blastomeres around the uncleaved yolky part. The blastocoel is absent. То есть egg of insect has in the center nucleus and thick layer of yolk around nucleus. On the surface on yolk, thin layer of cytoplasm without yolk. It's polylateral central-lateral egg. Cytoplasm with yolk does not divide, and divide only nucleus and parts of nucleus migrate on the surface of yolk, and from cytoplasm without yolk form a lot of small cells. And you receive periblastula. Periblastula has blastodem from equal small blastomeres, and in the center will be yolk. Cytoplasm with yolk, which does not divide. So blastocoel is absent. Mammals and human. Egg is untitled egg. Type of cleavage is a complete holoblastic cleavage, but uneven, blastomeres of different size and asynchronous. Type of blastula is blastocysta. At the egg cell stage of embryo, the cells form tight junctions and compact mass. At the transition from the 16 cells to 32 cell stage, the cells separate into two masses. The inner cell mass, embryoblast, is developed into the embryo, and the outer cells, the trophoblast, which will be parts of the placenta. So, blastocysta consists of two structures: outer layer, trophoblast, which will form placenta, and inner cell mass, embryoblast, which will develop into the embryo. It's electronic micrograph of blastocyst of human. And you see process formation of blastocyst. Next stage, gastrulation. This after cleavage, embryo, which called blastula, undergo gastrulation. Gastrulation is the process of formation 2 and 3 layer embryo, called a gastrula, or is a process of formation 3 embryonic layer, firstly ectoderm and endoderm, after that mesoderm. То есть blastula transform into a gastrula. The blastula develops a whorl in one end, and cells start to migrate into the whorl. This forms the gastrula, characterized by cell movement. Blastocoels is gradually disappear and a new cavity is formed, gastrocoel. During gastrulation, 3 germ layers form. The inner germ layer is the endoderm, the outer layer the ectoderm if early gastrulation, and after that the middle layer forms mesoderm. types of early gastrulation, то есть types of formation ectoderm and endoderm. The first types invagination. This type is typical for coiloblastula or amphioxus. Invagination blastoderm located on the vertical pole inside blaster coil and forming the inner layer, the endoderm, and the outer layer, the ectoderm. And you see the process. On the vertical pole, blastoderm inside blaster coil and form the inner layer, endoderm. Cell. The outer layer of outer layer is called ectoderm. And new cavity gastracoil or attenteron. Next process, epibolia. Type of early gastrulation is typical for amphiblastula of amphibians. Micromeres of animal pole of blastula divide faster macromeres of vegetal pole and overgrown them, forming ectoderm. And you see this process on this slide. Delamination. This type is typical for discoblastula of birds. Blastoderm stratified into two pieces, the inner endoderm and the outer ectoderm. And immigration. This type is typical for discoblastula of birds too. Some blastomeres of the blastula can migrate into the cavity, you see what on the second picture, and form the endoderm. So you must remember 4 types of early gastrulation: invagination, epiboly, delamination, and immigration. And last state of gastrulation, formation of mesoderm. The first method, Teloblastic method, is typical for invertebrates. You see gastrula, blastopore, and gastracoil or attenteron. On the body between the ectoderm and endoderm, but on this region, a group of cells called teloblasts separates. Thus you see teloblasts on this region. They divide and fill the space between ectoderm and endoderm, forming the mesoderm. This method of mesoderm formation is found in invertebrates. Next method, enterocoel method. The endoderm, this you see gastrulate again, blastopore and gastrocoel. The endoderm, only endoderm, forms protrusion towards the ectoderm. This protrusion separates their cell divide and give rise to the mesoderm. This method is typical for lancelet or Amphioxus. То есть mesoderm of Amphioxus is formed from endoderm. And third ways of formation mesoderm formation by migration of ectoderm cells through the primary strip. And you see embryo of birth. Embryon-like disc on the surface of yolk, its yolk. In the center of embryo, you can see primary streak. And cell migrate under ectoderm and line up on either side of the notochord on this radial notochord form to give rise to the mesoderm. This mesoderm is formed by migration of cell from ectoderm through the primary strip or primitive streak. In next lecture, we will begin with neurulation. So, today we consider next question. Pre-embryonic period, gametogenesis and fertilization. And parts of embryonic period, it's cleavage and gastrulation. Today our lecture is over. Thank you for attention. I see you later. Goodbye today.